Good morning. Thank you everyone for attending our 5th annual Chardon Genetic Medicines Manufacturing Summit, which again is a virtual format this year. My name is Keay Nakae. I'm one of the senior healthcare research analysts at Chardon, where our vision is to help identify companies with high potential for investment returns based on creating real value for society through disruptive innovation. Our compliance department has asked us to read this message, and this is effective for the whole conference. Ideas and recommendations presented are solely those of the presenters and not Chardon. Presenters have been advised not to discuss any confidential or material non-public information. Additionally, any of our conference speakers may have investments or other interest in the names that they discuss. With that out of the way, it's my pleasure to introduce our first guest from Lonza, Senior VP, Head of Cell and Gene Technology, Alberto Santagostino. The format for this session is roughly 40 minutes of fireside chat. If any of our audience attendees have questions during the session, feel free to type them into the question box under the video player, and we'll try to ask them on your behalf. So with that, let's get started. Alberto, welcome. Thank you so much for joining us. Could you provide a couple? Yes, thank you, Alberto. Could you provide a couple minutes of introductory comments about Lonza for audience attendees who are new to the company?
Very much happy to do so. And thank you, Keay, for the invitation. Let me just run through a few slides just to give context, but then I very much look forward to the meat of the discussion and to the Q&A. So for the general interest, Lonza is a quite broad organization that is serving the pharmaceutical industry quite widely. And as you can see, in terms of sales and employee, we are probably one of the most relevant players in the servicing of the pharmaceutical industry, definitely so from the CMC, technical development and manufacturing standpoint. As you can observe on the right, we are addressing virtually every modality. And I am representing the Cell and Gene Technology one, where I am the leading person in the representation of the company. We have services, so we provide CDMO services, and we have products that address multiple needs in those areas. Let me just run through you just a few of the elements that we believe to be important. We look at the world in 3 segments. Nothing new, nothing surprising. Autologous, allergenic, viral vector. What is interesting is that these sub-modalities are independent words from each other. With autologous, with a patient is one batch with all the natural complexity of manufacturing that comes with it. Every patient is a batch record. A lot of complexity with supply chain. Allergenic, more off the shelf, but definitely more tight regulation and more complexity from a CMC technical standpoint. And viral vector, which is a completely different modality from cells and allows to go really into genetic modification with importance controls that need to be in place and with the different modality of viral vectors that are there. In mind, these 3 different modalities, we are serving them all, but the very important stepping stone is whenever we are looking at supporting a client, we start from process development and supporting really the definition of a process that is manufacturable and can be industrialized. It really needs to start from having a process that when it's going to be scaled up and the numbers are going to become big and we are going to get toward commercial supply, the process is robust and actually can be performed effectively. The other important element regarding us is that we have one of our facilities in Houston where we have a significant establishment both in terms of viral vector and cell therapy. And now, as of a few months ago, it's officially certified for commercial manufacturing. This is something we are particularly proud of because there are not many facilities in the world that are authorized for the commercial supply of cell and gene technologies. The journey to stay on top of the regulation and continue being ahead of the game, it's important. And so we will continue working in improving and making sure that our standards are going to be ever increasing. But we are particularly happy to now have brought the first commercial product on the market. Also important to note is that our Nectar Cell and Gene Technologies is spread around the world with sites in the United States, in Europe, and also in Asia. Other elements is that Lonza is not only doing contract service and development, but we are also providing process technology. One of the things we are mostly proud of is actually the Cocoon, which is the next step in automation and industrialization of autologous manufacturing. You may know, but we have treated already the first patients with this Cocoon device, and we are currently running out the device in terms of allowing this to be accessible to whoever is interested. Other aspect is definitely the focus on industrializing viral vector, moving from 2D to bioreactor. Now the standards of 2,000-liter bioreactor is becoming the norm, as much as a focus on exosome that will be the modality on the future. Nevertheless, we also tackle, as I said before, the allergenic production with a particular focus in growing the volumes of patients that we can treat out of a single batch, as much as really putting a big emphasis on the GMP tissue sourcing, which is revealing one of the biggest headaches for all the allergenic companies that are approaching to market. Proper tissue sourcing need to be done in a very attentive and compliant manner, and we put particular focus in making sure that this gets done right. Finally, two final aspects. We really believe in the assays and in the characterization of the product. So we have been particularly investing in developing services regarding assay and characterizations, as much as we believe that a proper regulatory management through the journey of the product is essential. And so we provide particular support and attention on the regulatory journey. I just wanted to touch on a few of the elements we take care of. I guess that many of you may know who is Lonza, but I wanted to provide a flavor on the key elements that are at the focus of the service and what we provide. But now, Keay, let's go into the meat of the discussion, which I think is going to be much more interesting for everyone.
Well, great. Thank you for that, Alberto. I think that given your positioning as one of the major CDMOs, let's start with maybe getting your assessment of the state of the industry, and then we'll drill down into some of the more specific things that you're able to do. But obviously, to everybody on the call, the cell and gene therapy industry is growing exponentially as more early stage companies identify novel CGT targets, and the demand for CGT development and manufacturing capacity continues to grow. This increasing demand has created a backlog as demand is currently exceeding supply. In your perspective, what's the state of CGT industry in terms of capacity to start with?
Yeah. So the good news is that, as you said, investment are going in the area and will continue going in the area for a very justified reason. Efficacy is just astonishing, and safety is proving always better by every new therapy that is getting brought forward. So I definitely see in the equation of supply and demand, the demand side to stay robust, because it's very much justifiable. On the supply side, what we are observing is that there is significant investment in getting capacity deployed, but I would look at it in two different ways. I believe that the most important aspect is that we also look at capacity in terms of qualified capacity. Much of the capacity that is getting deployed today is intended to be for clinical stage or for supporting early phase. And many of also the commercial capacity that is getting deployed, it has challenges in getting staffed with appropriate and capable senior employees. Maybe the real aspect is that we probably should look at capacity not much in terms of square footage, but more in terms of people capable to actually run the operations. And this aspect, if anything, is the one that I see much more constrained in the industry. In terms of square footage, the amount of capacity that is getting deployed makes me even believe that not too far out in the future, there could be some sort of clean room overcapacity, but the big focus should stay on people, as we have seen recent moves of players divesting capacity.
Well, great. So let's touch on some of these things that you've mentioned. You know, compounding the capacity constraints is that the manufacturing of CGT products is complex. You know, many have stated that the current need for manufacturing innovation is similar to the evolution that occurred with the introduction of monoclonal antibodies, which themselves were much more complex than small molecules. Fundamental principles that underpin the success of MAB manufacturing innovation
was a solid understanding of the product biology, its critical quality attributes, and then designing processes to control for these, and then planning for analytical studies and tech transfer required to advance these next generation processes into the clinic. So when we think about cell and gene therapy, in your view, where is the industry first in its understanding of the biology of cell and gene therapies, given the diversity and variety of the different approaches? You mentioned autologous and allergenics, cell therapies, AAV, lenti, and and other vectors.
You are very much correct. And if anything, this is taken to the next scale because just almost making a provocation, I could say that the production of monoclonal antibody, it's easier in comparison because at the end is a sort of cookie cutter. You always have a producer cell line and then you're producing a monoclonal antibody. It almost doesn't matter what's the monoclonal antibodies for, but the unit operations for manufacturing the very same are quite consistent. Opposite wise, at the stage we are in the industry now with cell and gene technology, every single product is tailored. Even if you look at CAR Ts, there is not one process that is equal to the other, even if this is maybe the one that is mostly converging toward the standard. But if I look at number of open process steps, the unit operation are used in the very same, and the length of the same steps are practically all different among all my customers. This implies that it's really tailor-making. And this is coming from two reasons, partly because we are at the infancy, we are at the still very much in the science and not yet in this industrialization. And so there are not yet platform or standard that can be applied across the board. So what do we do is that we both observe that many players, and I don't want to put anyone on the spot, but later stage players that did not invest sufficiently in product characterization or process development do face challenges at the interface of going commercial, or more likely so, we observe an increasing desire and focus in really investing in the CMC technical part of the development to create a process that is robust and can survive the scale up and the commercialization. The good bad news is that at this stage, there is not any real platform, so a proper, diligent, and effective process development need to be done, and this is where we put a lot of accent. But also, we can envision that in the coming years, more so, there will be platform or at least a standard that will be deployed and made available. So we are just in the midst of the journey of industrialization, and we need to live with this ambiguity and uncertainty.
Yeah, so given all this complexity, the CDMO model is very much at the heart of CGT manufacturing compared to traditional biologics. How has Lonza shifted its business model to support the needs of small biotechs focused on CGT? You mentioned some comparability issues. We've got the need to plan for scale up early on. So talk about how you've shifted to help the small companies to be successful.
Yeah, that is a very, very important and good question. So the reality is that we had to make an acceptance and a change in our business model because we had to move from what was a standard offering, like in monoclonal antibodies, we have a GS system that we know is reliable and dependable, and we can plug virtually every recombinant protein into that system. Moving into cell and gene technologies, we had just to admit that we need to go into a mass customization exercise where, can you still see me? Because I don't see anymore myself.
I still hear you and see you.
Okay, sorry, something funny must have happened with the connection. So no, I just wanted to say that we moved into a mass customization exercise rather than pure industrialization. What does this mean? That we really need to take our client from the beginning and help them into developing their process to the proper standard. Obviously, there are different discussions. There are clients that are very much interested to go first in human and proof of concept because they want to test out if their technology works, and so they are more focusing on speed. And then there are other clients that are more focused into really developing a stable platform. It's never black or white, it's always a mix of gray, and we need to be able to understand what are the real needs of our customer. Typically, the two things are both needed. So there is the need to get relatively fast into a proof of concept, but at the very same time, in the background, there is the need to really develop a robust process. What do we do is that we accompany our customer through the journey. We are far from perfect, but in this moment in the industry, I guess that is a general complexity to cope with these challenges. But we are relatively proud about the job we have done so far.
So the manufacturing needs of the small biotech will change along their journey to commercialization. So how does Lonza support them along this journey, starting first with the move from the lab bench to supplying product for IND enabling and human clinical studies, and then supporting, commercialization scale up?
Yeah. So the very important aspect is that many of those biotech, the great share of those biotech, are gonna be aiming at either a sale or a licensing to bigger pharma. And in that very interface, there is going to be an important moment of truth in which the process will be looked at as much as the clinical results will be looked at. And in that moment, there will be an analysis of understanding if what is at hand is actually viable, both in terms of cost, because those therapies are not inexpensive, and in terms of robustness of the process. You probably know around several autologous products that are reaching the market that really struggle to meet release criteria, and this is creating significant headaches for the company that are currently commercializing those products. And this is really the complicated exercises. More and more, what I can tell you, I'm observing is that there is a desire to invest into real and appropriate process development early on. And it's not rocket science often, it's just down to simple things like choosing raw materials that have a GMP background, making sure that as many possible process steps are actually closed to expose less to sterility issues, or things like making sure that every given manufacturing process stays within a number of hours that is achievable and possible to be served within a work shift of a blue collar.
Well, you mentioned that the eventual commercialization may be done by a partner, but other companies are looking to make at some point a make or buy decision because they were choosing or because they have a platform that could offer multiple products. So some of them choose to do a hybrid strategy. So from your perspective, in terms of risk benefit, which task can the emerging biotech company take on, and which are better left in the hands of the CDMO to perform?
Yeah, it's a very good question, and there is not a one answer to the matter, because the choice of keeping internal or externalize or having both, it's very intimate in every company situation. For sure, the decision of internalizing and building its own manufacturing facility is definitely capital intensive and also knowledge intensive. The talent is scarce, the capital less so. And so it's a big investment in terms of management, a share of attention, and also risk taking. On the other side, there is the upside of having control, and maybe if the company has some specific technology manufacturing that is considering a core competence, it could be justified. What we observe is that it's a bit of a mix. We have companies that desire to leverage us in terms of avoiding capital investment, leverage us in terms of access to knowledge, leverage us in terms of optionality in case their internalization plans do not work. So I would answer to this question that it's really depending on the specific context of every client, what is the right choice. Obviously, the way that we set up things is that there is always an option for customer to internalize or tech transfer out. And what we observe is that in multiple cases is either they stay with us, or if they internalize, they keep still the optionality to continue manufacturing with us, probably as a de-risking factor.
Okay, so let's drill down to some of the specific challenges in just manufacturing the product itself. You mentioned your Cocoon system, which is a closed system, which offers a lot of advantages. Can you expand on what Cocoon currently is able to achieve in terms of reducing validation requirements or informatics or other efficiencies?
Yeah, very happy to do that. So the Cocoon has been invented and created after a vision and a reality. If you consider Autologous, it's a complete change of what is the normal manufacturing paradigm of pharmaceutical companies. I remember to have seen a company that has 2 years of inventory of finished good inventory. With Autologous, you, from the raw material to the dosing of the patient, there are 15, 20 days and a pretty tight supply chain. Very complex, very just-in-time manufacturing approach, which has also shifted the relative ratios of the incidence. Typically, manufacturing is the biggest cost incidence, while QC, QA are just a minute part in terms of patient cost. In reality now, with the Autologous, you can almost have the rule of 1/4 is raw material, 1/4 is manufacturing, 1/4 is QC, and 1/4 is QA in terms of cost. Cocoon is able to reduce significantly the cost of at least 2 of these factors. One is the manufacturing by creating automation and standardization into the manufacturing approach. So cutting off 1/4 of the cost I was mentioning about, and also the aspect of information management and quality assurance, because we are going into an electronic batch record and into an automation system. These allow, per se, to significantly decrease cost, but even more importantly, enable scalability. And so it's a very easy way out to scale out the process. It doesn't require particular training of people. It doesn't require anything more than taking what works in one Cocoon into 50 or 100 of them. So there is an ambition of scalability, reliability, robustness, and definitely cost cutting by trying to address some of the key pain points of the current Autologous setup.
Okay. Specific to autologous cell therapies, is there any other technology advancement that could have a significant impact on reducing the manufacturing cost?
So specifically on Autologous, I believe there is going to be one fundamental change that is related to the way we do gene modification, genetic editing. As of now, lentivirus has been predominating in the market. It is a significant share of the cost and an even bigger headache in terms of supply chain reliability. My personal view is that we are going to move away from lentivirus transfection, and we will increasingly see non-viral approaches like mechanoporation or electroporation. This is also one of the reasons we have been significantly investing into a device in Lonza that is called the Nucleofactor, which is an electroporation device. We have already several customers that are using that device, and what we are observing is that it is more dependable. It kind of delivers always the same consistent performance. It's less variable compared to the lentivirus, and on top of that, cuts away a lot of the supply chain and the cost related to lentivirus.
Okay, and how far off is something like that from being, being able to be deployed?
Well, there are customer and players that are already in late phase 2 and entering phase 3, so it's about to happen. Okay,
great. How about other aspects of manufacturing? Let's talk about recombinant AAV. What types of shifts are you seeing in let's say moving from suspension to adherent, or the use of baculovirus cell lines?
Yeah, I think the most important aspect is the scale up in this case. We are by now practicing the 2,000-liter bioreactor as a standard in most of the process we are currently serving. And that is already reaching a scale that with today's titers and yield, it's already delivering a reasonable cost of goods. But the journey will just continue in increasing the scale and increasing the technical performance in terms of titering yield. There are many avenues to work that out. Some of them are purely technological. Some others are more related to how operations are run. And so we, in that part of the viral vector production, we will see the repeat of what we have been observing in recombinant protein manufacturing. From a purely manufacturing standpoint, the technology are fairly similar. And so we can leverage a lot of the knowledge that we have built into that previous field.
Okay, so what other current manufacturing bottlenecks, either upstream or downstream, do you see as addressable right now?
Yeah, I mean, we talked about autologous, we talked about viral vector. Let me maybe touch on the allergenic aspect. Allergenic cell therapy is maybe 1 or 2 or maybe 3 steps behind compared to viral vector manufacturing in terms of moving from a 2D to a bioreactor setup. And that is something that we are more at the beginning than compared to viral vector. So I would expect a significant step up in terms of bioreactor and scale up in allergenic manufacturing, which is also an area where we are particularly focusing on with some of our customers. That is one of the aspects I would see to advance quite significantly, as much as I would see the sourcing of the starting material to be an increasing area of focus, which needs to be done in a proper manner. We observe that many later stage products now are in the need of really establishing a proper tissue acquisition to really be effectively compliant for the commercialization phase.
So just expanding on that last point you made, in terms of future innovation, how much is dependent on new technology? How much is dependent on just being more creative in how you actually do the processes?
Yeah, we refrain to comment on the basic biology or on the product because that maybe is not the core of my expertise. But if we just look at CMC technical development, we are just at the beginning of the journey. And as it always happens, is a 3-dimensional improvement. One improvement is pure basic operations. As of today, many things has been run like in a university lab, and there is the need of stepping up and going toward a more industrial process approach. So the process just need to become easier to manufacture and easier to scale up. And often those things are super trivial. It's about matching a proper work week. It's about using equipment that is more robust and resilient, and simply manufacturing schedule and things that are simple as that. The other avenue is definitely also the technological advancement. I gave you the example of the Cocoon. I gave you the example about working on titering yield on adenovirus or lentivirus. And that aspect of technology is also pretty foundational. As you've seen in recombinant protein, the titer and the yield has increased over time consistently. And so much so we need to do into cell and gene technology. And the third one is just going to be simply on the raw material supply, because at the end, the current input cost of raw material is significant. And as the industry will mature, we will be observing that those costs are going to decrease and come down. Nothing different that we've been observing in the previous situation, like where to produce monoclonal antibody or recombinant protein, there was always serum-rich media. And now having a recombinant protein manufacturing that uses serum, it's almost an exception. So we will see things like those of these 3 avenues of improvement that will come about and improve the current situation.
Great. Well, another aspect that certainly impacts what you're able to do on the manufacturing side is how up to speed the regulatory agencies like the FDA, EMA are with the latest and greatest manufacturing approaches. So in your opinion, where are they in terms of being up to speed? You know, what do they still need to get comfortable with?
Yeah. So we just have to admit that FDA and the other regulatory agency has been extremely on top of this new technology, and they have been extremely capable in really driving both adoption and compliance. On an initial phase, as it's natural, regulators try to enable an industry, so to have more allowance in terms of more loose characterization acceptance criteria, and really acknowledging the value to patients versus equivalent stringencies that were applied in other modalities in the industry. And what we are observing, as it naturally should happen, the regulators' expectation in terms of compliance and appropriateness of manufacturing, CMC standard, characterization, and just the way of doing things is becoming progressively more tight. And we just as an industry should acknowledge the incredible help that regulators has provided to enable the industry and get to a rapid start and blossoming, as much as now we need to collectively embark in a journey of everlasting improvement. And this is what we are trying to do in as much as humble possible way to continue working on improving our processes, improving our compliance, improving our delivery to meet ever-increasing standards and appropriateness of compliance. But I can for sure tell you that the regulators are very much on top on both of the mainstream cell and gene technology, as much as very curious about the new technology. So they are at times more forthcoming than companies or CDMOs.
Great. You mentioned earlier the lack of qualified workforce talent is a current capacity constraint. So what is Lonza's strategy for attracting, developing, and retaining qualified people?
Yeah, I guess that this is a challenge for everyone in the industry. What we are trying to do is that we are trying to find a mix of good available talent, as much as hiring people that then we train personally in the job that we want them to do. Lonza is for sure a great school, so it's a beautiful place to both get started in the industry and continue the journey. The current growth rates that we have in these modalities such that opportunities of career are significant. And so there is definitely the possibility to have a journey inside the company, and then the intellectual interest of being exposed to dozens of new products every year is just sometimes extremely engaging. So compared to an originator working in a CDMO, and particularly in Lonza, if someone is passionate around the CMC technical development, there is no better place, because it's not that we have a portfolio of 1 or 2 products, we are in the hundreds. So it's easy to get exposed to a variety of learning and scientific interest that is difficult to be matched in other parts. But for sure, the market is very hot, so I don't deny that we struggle to find talent because we are hiring in the hundreds every year. And so if you know anyone good that wants to join us, just send the CV. I'm going to be very happy to review him and probably offer him a job.
Great. Alberto, the development of available COVID-19 vaccines has been accomplished at unprecedented speed. What do you believe are the key things learned that can be leveraged in the manufacturing of CGT going forward?
Yeah. So I think this situation is a bit of an exception in the sense that not every product can be developed as a BioNTech or a Moderna or any other of the vaccines that has been put forward, because the level of investment, the level of effort, the level of commitment that is put on one drug to do such of a speedy development is just unsustainable to do it in a broader spectrum of things. But why not? It could be justifiable on certain product and indication. So the way that we have been working on enabling these therapeutics is fundamentally dedicating our best people and investing in a per unit of whatever, could be engineering, could be process development, could be manufacturing, 3, 4, 5 times more than we would have done in any other place. So it's feasible, and I guess that it should be conceivable, but I guess is not possibly practicable everywhere for every product.
Okay, if we look out 5 years from now, what do you think will be the biggest changes in manufacturing?
It's very difficult to predict and anticipate what will be, but one thing is pretty clear in my mind. Cell and gene technologies are so transformative and are able to address unmet needs that cannot be addressed anywhere in any different manner. So there is going to be an incredible pool from everywhere in the industry to make those drugs and those approaches universal, and not only for niche diseases or things that are exceptions. It will become eventually mainstream. And we can envision multiple things like having individuals being screened for all the possible genetic diseases that they are carrying and getting treated for them. We can envision even regenerative medicine that we didn't really discuss here. The potential of cell and gene technology as a regenerative medicine is all to come, but is going to be extremely important in terms of ensuring healthy, long lives. And so all those things are going to come about, that there will be an incremental patchwork of innovation that collectively will just make a phenomenal change. So it's particularly difficult to anticipate any single one or two items that will make an enormous difference. But I can see that collectively, this is going to be an astonishing revolution that cannot be anything but exciting. And for sure, it's going to happen.
Yes, we agree. So let me ask you one final question, just to summarize everything you've talked about. How does Lonza differentiate itself as the preferred CDMO partner?
Yes, that's also a very important question. What we are trying to do in our best manner possible is try to stay faithful a little bit to our image and to our and what we stand for. So trying to be as reliable as possible, as committed as possible, focused on commercialization. So we are maybe a little bit more clunky and we are maybe a little bit more complex, but we really try to really bring forward this concept of industrialization and carry customer from the early stage till the market. As I told you at the beginning, we are far from being perfect. It's difficult to do so in an industry that is not yet industrialized, but is very much tailoring or mass customized, and is very difficult to do so in an industry that is growing 30, 40+% a year. So there is a lot of turmoil. We also kind of experience that, but we try to keep at least a very straight light around the objectives of one after the other, bringing our customer to the end result of bringing their product to the market. And that is what we really try to live for and stand for.
Okay, well, very good. We've come to the end of our session here. I want to thank you so much, Alberto, for joining us today. And with that, we'll end this session.
Very much thank you and appreciated the invitation.
Thank you.